My name is Dr. Chelsea Bennice. I am a research scientist for Florida Atlantic University's Department of Biological Sciences. I grew up in Ohio near Lake Erie. I have always loved the water. Growing up, my family would take vacations to Florida where I would get to spend time in the ocean. Today, we are visiting Blue Heron Bridge, Phil Foster Park. This is a unique spot within the Lake Worth Lagoon in Riviera Beach, Florida. I've been diving Lake Worth Lagoon for over 7 years, and I still get excited to be underwater. I first got interested in octopuses when I saw their camouflage. Not only can an octopus change its skin color in less than a second, it can also change its skin texture from smooth to bumpy. On my research dives, I'm usually bringing multiple science tools to help me answer my research questions. Today, I brought my camera, quadrats, an Octopus Monitoring Gadget, or what I like to call the OMG, my floating lab, and my dive buddy. The OMG is a camera I leave underwater for 24 hours to record the octopus's activity time. I think of it as a day in the life of an octopus. I also get to see the other animals in the area, what the octopus is eating, and any interactions the octopus may have with other animals. First, we entered the water and started the dive by searching for an octopus. There are 2 populations of octopus species that are found in the Lake Worth Lagoon. I like to call them underwater neighbors. They live pretty close to each other, but are able to coexist and not fight for resources because they use different types of substrate to create their homes, have different diets, and are active at different times of the day. Today we found the Atlantic longarm octopus. This is a small species of octopus with its mantle, the sac behind the octopus's eyes, only growing to 2 to 3 inches. But it has super long arms, about 6 to 8 times its mantle length. We saw multiple octopuses on this dive, and this one was out searching for food. So I grabbed my camera and started recording the foraging behaviors. Next, I used my square PVC pipe frame, called a quadrat, to lay over the area of the octopus's home, also known as the den. After the quadrat was in place, I took photos of the octopus's den and surrounding habitat. Since I know the area of the quadrat, I can look at these pictures in a computer software program and quantify how much of the octopus's den is made up of sand, rock, shell, flora, and fauna. While the octopus was still out of its den, I used a small basket to temporarily collect the octopus and carry it to my floating lab. I'm able to ascend safely with the octopus because I am diving in shallow water. Once I was at my floating lab with the octopus, I gave the octopus a belly rub. I used a sterile swab, like a Q-tip, to collect the mucus and bacteria from the octopus's mantle. After this, the octopus was released back to its home. I'm interested in the role bacteria play in octopus health. To understand if octopuses have a unique community of bacteria called the microbiome, I also took a water sample and a sediment sample. I will take all 3 sample types back to the lab and compare the bacteria present in each sample. Being underwater is my happy place. The blue ocean water is soothing and it makes me feel relaxed. It's nice to get away from everyday noises and be underwater where it's quiet, or to simply listen to the shrimp snapping. My first introduction to marine biology was in high school. After learning about marine environments and the animals that live in the ocean, I was hooked. I read all the marine biology books. I even started my own saltwater aquarium in my bedroom. My first experience seeing these tropical fishes in the wild was on a snorkel trip to the Virgin Islands. This is when I knew I wanted to find a career where I could spend my time in the ocean. My message to aspiring female scientists: go for it. Turn your dream or passion into a career. It's a lot of hard work, but it's worth it.